Hello and welcome back to today's session. Today we are focusing on a critical enzyme that protects your most numerous cells, the RBCs. We are discussing glucose 6 phosphate dehydrogenase deficiency, famously known as G6PD deficiency, the most common enzyme deficiency in the world. Think of RBCs as a submarine responsible for transporting oxygen. The cell is under constant attack from oxidative stress, that's our enemy torpedoes. The enzyme G6PD is the internal shield generator and repair system that keeps the submarine safe. G6PD deficiency means the shield generator is faulty, leaving the RBC vulnerable to attack and destruction. So, the pentose phosphate pathway, HMG shunt pathway, is the RBC defense hub, and G6PD is the first and rate limiting enzyme in this pathway. So, G6PD takes glucose 6 phosphate and converts it into NADPH. It converts it into a product, generating NADPH in the process. So, NADPH is the energy charge or energy needed to power the shields. It is absolute currency of the protection inside the RBC. NADPH then passes its reducing power to glutathione reductase, which regenerates glutathione from its oxidized form. So GSH, that is glutathione, reduced glutathione, is the molecule that directly neutralizes harmful reduced reactive oxygen species like H2O2. So here our activated form, that is the reduced glutathione, is the active shield. The NADPH charge is needed to continuously produce the shield, allowing it to intercept and neutralize our oxidative torpedoes, that is our ROS. So in G6PD deficiency, the RBC cannot produce enough NADPH, leading to oxidative damage. So it's almost inherited, like genetics. G6PD deficiency, it's an X-linked recessive disorder, meaning it's far more common and severe in males. The gene that makes the shield generator is defective. So the state, it's most of the time RBC operates fine because the level of oxidative stress is low. In case of hemolytic attack, like the triggering torpedoes, if at all during a hemolytic crisis, as in a massive RBC destruction, only occurs when the RBC is exposed to high oxidative stress. So during this period, this stress is activated by fava beans, like favism. It contains certain compounds that trigger ROS production. Or infections, immune response generate massive ROS. Oxidant drugs like sulfur drugs, primaquine and aspirin, so these generate massive amount of ROS too. So these triggers are the enemy submarines firing a massive volley of torpedoes. Since G6PD shield generator is broken, RBC is helpless. And when the shields fail, the oxidative torpedoes hit the submarine, leading to characteristic signs of RBC destruction. That's hemolysis and Heinz bodies. The ROS attack and denature the cell's hemoglobin. The denatured hemoglobin clumps together and adheres to the cell's membrane. The oxygen tank, that is the hemoglobin inside the submarine, is damaged and forms rusty clumps, that is our Heinz body, that sticks to the walls. When these damaged cells pass through the spleen, specialized macrophages take a bite of the RBCs to remove the Heinz bodies. So the spleen security force, macrophages, detects the rusty clumps and takes a bite out of the submarine's hull. The damaged cells are destroyed, leading to a sudden drop in RBC count or hemolytic anemia. Clinical signs is predominantly we see jaundice due to massive bilirubin release from RBC destruction, dark urine and fatigue. So remember G6PD deficiency as the faulty shield generator of the red blood cells. It leaves the cell unable to produce protective NADPH. When exposed to oxidants, the RBC suffers maximum damage, leading to Heinz bodies, bite cells, and acute hemolytic anemia. So this key is to avoid the oxygen triggers. Now let's get back to another deficiency, that is our pyruvate kinase deficiency. Think of glycolysis as a short 10-step energy express assembly line that breaks down glucose. Pyruvate kinase is the final cashier at the end of line. It collects the last big paycheck for the cell and dictates whether the entire process speeds up or shuts down. So pyruvate kinase catalyzes the third and final regulatory step of glycolysis right before the pathway is complete. Pyruvate kinase transfers a phosphate group from PEP directly to ADP, producing two products, pyruvate and one molecule of ATP. This is the final cash transaction. PEP is the high value security check and PK is the cashier that converts check into readily usable cash, that's ATP. Since this happens for both halves of glucose molecule, it generates two ATPs per glucose. So this step is an example of substrate level phosphorylation. PK is highly regulated enzyme ensuring glycolysis only runs when needed. Feed forward activation. It's activated by fructose 1,6-bisphosphate, a product generated earlier in the pathway. So this enzyme is the incoming order slip. When the cashier sees a huge stack of orders from upstream, that's when fructose 1,6-phosphate is elevated. It knows production is high and speeds up the checkout process to match the flow. Allosteric inhibition. It's inhibited by high levels of ATP and alanine, a product of pyruvate. So ATP is the full bank account. If the cell's account is full, the cashier closes the line. Alanine is the warehouse overflow signal telling the cashier to stop producing more raw material, that's pyruvate. So, PK deficiency is the second most common enzyme deficiency causing hemolytic anemia after G6PD. Hemolytic anemia, so RBC energy failure. The root cause is due to the red cells rely exclusively on glycolysis for their ATP. They lack mitochondria and cannot use the TCA cycle. The red BC is mitochondria-free factory that only uses the energy express line. If the final cashier, that is PK, is missing or slow, the RBC cannot get its final ATP payment. Lack of ATP causes the RBC to fail at operating its crucial ion pumps, leading to dehydration and rigid cell membranes. The stiff cells are prematurely destroyed by the spleen. Chronic hemolytic anemia, splenic destruction of RBCs, and or it's also due to the engulfment of Heinz bodies by our macrophages, and a buildup of upstream glycolytic enzyme intermediates. So these are the clinical signs. Consequences. So when the PK is deficient, the intermediates just upstream pile up. One of these is 1,3-bisphosphoglycerate. It's shunted into a side reaction to make 2,3-bisphosphoglycerate. So the stalled checkout line causes customer to move into the side kiosk. And high 2,3-BPG lowers hemoglobin affinity for oxygen and enhances oxygen release to the tissue. This is a beneficial compensatory mechanism. So patients with PK deficiency can often tolerate surprisingly low hemoglobin levels because of their RBCs are so good at dropping off oxygen. So remember pyruvate kinase as the final cashier of glycolysis. It's the critical step that generates final ATP. Its deficiency specifically cripples the red blood cell factory, causing hemolytic anemia, but paradoxically this leads to an increase in 2,3-BPG, which helps the body to improve oxygen delivery. So that's it for today's session. Keep on practicing.